Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Biomodels, LLC, I'd like to welcome you to Tailored to Translate, Custom Preclinical Models for Advancing Diverse Therapeutic Strategies in Oncology. My name is Elizabeth Lamb, and I'm the host and the moderator for today's event. Now I'd like to introduce our presenter for today. He is Benjamin G. Cuffo, PhD, Principal Scientist in Oncology for Biomodels, LLC. Welcome, Dr. Cuffo. The presenter ball is yours.
Thanks very much, Elizabeth, and thanks for everybody joining with us today. I'm really happy and excited to be with you today to tell you a little bit about our oncology program here at Biomodels and how we can leverage and customize preclinical models to advance diverse therapeutic strategies and to enable improved translational success. So our topics for today, I'm just going to go through really quickly. I'm first going to introduce you to our company, Biomodels. I'm going to talk a little bit about the oncology pipeline and advancing novel therapeutics in oncology and some of the challenges involved there. Go over the preclinical oncology model toolkit quickly, and then spend most of my time today talking about custom models for these diverse therapeutic strategies. And I'm going to go over a few case-by-case sort of studies that we've had the opportunity to conduct here and cover sort of a lot of different things within the context of these studies. So the first will be a microbiome and immuno-oncology, looking at checkpoint inhibitor combination therapies and some of the ex vivo assays we can perform here. Next would be modeling standard of care chemoradiation regimens in the preclinical mouse models, and also relapse, looking at orthotopic sites and the use of bioluminescent reporters. We're going to look at our panel of bioluminescent PDX lines. This in combination with metastatic models and some additional clinical recapitulation in the models. We're going to look at cancer treatment toxicities in tumor context and simultaneous modeling of related comorbidities. And also look at some of the next generation models that we're working on right now, developing, including triple-humanized models, looking at considerations of scheduling and modeling of tumor microenvironment in vivo. So quickly, Biomodels is a preclinical contract research organization. We're located in Watertown, Massachusetts. We're bordering on Cambridge. We're dedicated to collaborating with our clients, which range from large pharma to small startups to academia, to bring new therapies to patients. We're in our 21st year. In 2018, we're founded in 1997 out of the Brigham and Women's Hospital. We're now an independent company. We design and execute translational models of human diseases and have enabled more than 50 drugs, biologics, or devices into patients over multiple indications to date. We have expertise in design and execution of cutting-edge preclinical models for oncology and cancer treatment-associated toxicities, referred to as cancer supportive care, inflammatory and autoimmune diseases such as IBD, colitis, GVHD, pulmonary diseases, cardiovascular disease, neurological diseases, and many, many others. Okay, so that was the quick introduction to Biomodels. Now into the overview of the cancer therapy development pipeline, which many of you will be familiar with. But just to go over really quickly, we are going to want to develop our novel therapeutic from concept to patient, and we're going to need to run through different steps in the discovery stage and also the development stage. So preclinical, we're going to identify targets, identify compounds or biologics or whatever the therapeutic modality may be. We want to establish activity. We're going to select, do clinical patient selection and test safety, and then before moving into human clinical trials. So to move into human clinical trials, we need to get approval from the FDA in the form of usually an IND, investigational new drug. And to do so, we basically only need from the FDA at the very minimum to show safety and some efficacy. So this seems pretty straightforward. You know, we know how to provide safety data. We can look at pharmacokinetics, find a safe dose, and assess toxicities. But really, the next step is where it becomes more complicated, and that's providing the biological activity data. And this not only stretches from the preclinical stage, but also into the clinical stage, and I'll talk more about that as we move forward. But we need rationale, we need mechanism of action, and we need efficacy. And for FDA approval following the clinical phase, we need substantial evidence of efficacy with acceptable safety. It's got to be greater than or equal to currently available treatments. And we need to have an appropriately defined patient population that is likely to benefit. Some of the challenges in doing this in oncology is that cancer is the most highly individualized disease indication, most likely, and predictive or selective biomarkers still remain elusive. We also need to provide adequate information to enable safe and effective use, and this really comes back to identifying the patients most likely to benefit in oncology. Another challenge is that the patient presentation and clinical treatment is going to vary widely from patient to patient. And you can see that some of these challenges do cause issues in oncology versus non-oncology therapeutic development. You can see in this graph on the right that oncology has lower levels of transitions between the phases than non-oncology therapeutics. And some of the reasons for these discrepancies might be, you know, insufficient preclinical development, difficulties in matching patients to treatments, and or design or execution issues in the clinical trials. With just a little extra preclinical development, for example, in this case, if we have a biomarker we can use for patient selection, we are able to improve our likelihood of success in transitioning from one phase to the next. So I'm just going to go through the clinical trial phases really quickly and talk about the reasons for failure. So in phase one, we're just testing very small groups of people, 2,100. We want to evaluate safety, side effects, and it's really the first in human phase. We want to determine a safe dose range. The primary reason for failure there is safety, usually off-target effects. So preclinically, how can we try to mitigate this? We can perform safety and efficacy studies in clinically recapitulative preclinical contexts. We can do preclinical dose ranging to predict efficacy safety margins in human. We can perform longer-term preclinical safety studies. We can do enhanced tox analysis of off-target animal tissues. For phase two, we're testing at the effective therapeutic dose in patients, usually about 100, 300 patients. We want to identify the patient populations most likely to benefit. Primary reasons for failure in phase two are either efficacy or patient accrual. So how can we mitigate these reasons for failure in preclinical modeling? We can establish confidence in the mechanism of action. We can utilize preclinical efficacy models that specifically test the mechanism. We can vet efficacy in increasingly challenging clinically recapitulative models. We can do efficacy in context, test multiple contexts and biologically accurate contexts. And we can try to learn from the non-responders to enhance patient selection. And just gather more data, establish non-invasive biomarkers. For phase three, we're really testing at therapeutic doses in larger groups of patients. We're testing in the clinical context in combination compared with the current standard of care. And it's the final evaluation of patient risk benefits compared to the standard of care. Primary reasons for failure here are, again, efficacy or non-scientific reasons. Preclinical strategies to mitigate failure in phase three, do preclinical efficacy testing in combination with standard of care, recapitulate clinical standard of care treatment schedules, ask how the standard of care treatment might impact efficacy in terms of how a a patient undergoing previous treatment might respond to a new treatment, and how the new treatment might work in synergy with standard of care treatments. And run models that account for off-target co-toxicities, potential co-toxicities with standard of care. And also look at rational preclinical models that model standard of care resistance for that. So this brings us to the 5 R's of therapeutic development success. We want to find the right target in the right tissue with the right safety, importantly identify the right patients, and have the right commercial potential. So this is sort of the sum up of this preclinical oncology pipeline, developmental pipeline. Why do we think more R&D dollars might be better spent with co-clinical or preclinical studies prior to or in parallel with clinical trials? We think ultimately we can save money, save time, decrease false negatives, identify patient populations most likely to benefit, establish meaningful endpoints, be able to test therapies in combination, which are more accurate, increase confidence when you're entering the clinical phase, and ultimately increase clinical success rates. Now, the oncology pipeline is filled today with really diverse therapeutic strategies, really diverse treatment paradigms, and this makes for a ton of variables in how they'll all work together and how we might want to assess these preclinically before going into the clinical trial. So we have different therapeutic modalities such as small molecules, biologics, vaccines, viruses, cell-based therapies, and microbiome modulating therapies. These can be administered locally, systemically, in a combination of the two. We want to select patients with biomarkers, genetics, patients that might require some preconditioning treatments. Will the therapy be administered as first-line therapy? Might it be a second line? Will it be a single agent? Will it be an adjunct agent? And all the combinations, the schedules, the regimens really play into how well the clinical trial will go. So we think that preclinical studies should be custom tailored to answer these specific questions. We don't want a one-size-fits-all study to answer a very narrow range of questions. We want a highly customized study to answer very specific questions and to have those questions answered specifically. And the preclinical oncology toolbox, the current time is a very wide-ranging toolbox. We have all sorts of different types of models, different cell types. These can be combined in many ways with different current standard of care therapies, and we can look at different primary endpoints. So just the main types of models that are out there, there's a whole host of in vitro models that model complex interactions in a dish. They're great for looking at biochemical, molecular interactions and pathways. We have genetically engineered mouse models that can be layered with various xenograft or tumor genesis models. Your murine models, your immunocompromised models carrying human tumors, and then up from there you have more humanized animals carrying human tumors and human immune cells. We have PDX models that are more closely coming from patients that often come with very detailed patient information, and also germ-free or gnotobiotic models. These are animals that are raised in a germ-free environment and can be colonized with microbiome that's either clinical or specific bacteria. And these can be, as I just said, combined with a whole bunch of standard of care therapies to look at combinations, and then primary endpoints can be matched to clinical endpoints such as survival, disease-free or progression-free survival, time to treatment failure. We can look at biomarkers and other readouts. So we like to think of these models sort of as a gradient of biological accuracy in the model, and we think that the biological accuracy of the model will correlate to the expected predictivity, translatability of the results in patients. So maybe, you know, at the very left of that, we might have some 2D culture models in a dish, which are very useful for some things, but are maybe not the most biologically accurate systems, all the way up the gradient to, you know, a really well-characterized PDX in a humanized animal that's really clinically recapitulative as much as possible. And using all the pieces of the tools in our toolbox, you know, we specialize here in designing custom models or working with you to design custom models or smart models that will consider all of these factors and take advantage of, you know, the mechanism of action, try to be as clinically accurate as possible and biologically accurate as possible, and really try to assess translatable endpoints. And in all of this, we do stress that you know, one study is great, but it's best to maybe do iterative preclinical modeling to really get you know, more repetition and also answer more questions in a well-defined way. So I'm going to turn now to some of these case studies. And the first thing I'm going to talk about is microbiome in immuno-oncology, looking at checkpoint inhibitor combination therapies and ex vivo assays. So just a quick introduction to the microbiome. So humans, like pretty much all multicellular organisms, are meta-organisms. We're comprised of both a host organism and thousands of species of symbiotic organisms, including bacteria, fungi, viruses. Our own cells are actually outnumbered by symbiotic microbial cells by a factor of 10, expressing greater than 10-fold number of unique genes than our cells do alone. Our immune system co-evolved to contain and control microbes, and as a result of this co-evolution, symbiosis with commensal microbes took hold, with certain microbes providing beneficial enzymatic and immunomodulatory functions for the host. So it's likely no accident that the highest numbers of immune cells are resident in the gut and the skin, and that's where most commensal microbes live. So it's a really tight connection between the immune system and the microbial population known as microbiome, and it's really a co-mediation of immune homeostasis. So basically, what the immune system is doing in response to these bugs is balancing inflammatory response to potentially dangerous pathogens with tolerance for symbiotic commensals or metacells. And the immune system is able to do this through a combination of antigenicity and adjuvanticity provided by chronic exposure to the microbiota. And recent papers in Science and elsewhere have really shown that this is impactful clinically. Patients respond differentially to checkpoint inhibitor therapy in these two papers based on the composition of their microbiome. So this really matters for efficacy and response. And so today, rational modulation of the microbiome is emerging as a promising therapeutic intervention. As a wide lens view of microbiome in immuno-oncology, really the influence of the microbiome should be controlled for in every preclinical oncology study where the mechanism of action relies or may rely upon the immune system. It's that much of a modulator, and you may see false positives or false negatives depending on the microbiome of your mouse. So mice should be sourced from the same vendor or barrier for series or repeat studies, and mice should be housed and handled under containment protocols that are constant, ideally individually ventilated caging, receive food, water that is consistent, not introduce potential perturbations to the microbiome that are not intended. And that's just controlling for a variable that is needs to be acknowledged in immuno-oncology. If we want to go ahead and try to translate the microbiome, modulate it to improve efficacy, we can use some of our preclinical oncology models in the context of microbiome. Some of the treatment paradigms there would be modulation of the microbiota with live bugs or drugs that will impact their population. We can use germ-free mice to perform fecal microbial transfers, or FMTs, where we actually colonize with clinical microbes and look at the education of the immune system. Or we can look at a variety of other treatment paradigms within this context. This is just a quick slide to show a bit of the barrier levels to keep the microbiome controlled. With conventional barriers, you're going to have cages that have no barrier, or enhanced containment protocols, which we use at Biomodels, have the animals are kept in HEPA-filtered cages. However, the cages might be open and exposed to the facility microbiome. So you're going to have control between groups, but maybe not control of the facility. The next level up, which we offer here as well, and we perform a number of studies of this containment protocol, is the specific pathogen-free, and that's where all of the cages are only opened in laminar flow hoods that are pre-sterilized, and so the microbiome that the mouse comes in with is the one it's going to have throughout the study, and it's going to be stable. A level up from that is our Gnotobiotic or germ-free facility, where the animals they come in germ-free, they're completely uncolonized with any microbes whatsoever, and we can put in anything we want, colonize them as a blank canvas, and they live in their lives in these bubble isolators. So this is just a look at our germ-free Gnotobiotic facility, which has been open for about 2 years and it's been very active. So what are some of the strategies we can use in terms of looking at microbiome? Probably the most simple one is to modulate an endogenous or healthy microbiome. So again, there's a lot of words on this page, but basically these kind of models can be layered to look at specific questions and specific readouts. But basically, these are models where the animals will come in and be kept at specific pathogen-free, SPF plus containment, and will have their microbiome controlled for throughout the study. So getting into the actual study, so here's a study we had the opportunity to perform where we looked at animals from different vendors, genetically identical animals from different vendors that ostensibly have a different microbiome. And we looked at how they responded in a B16F10 melanoma model to a suboptimal dose of anti-PD-L1 checkpoint inhibitor therapy. And this was replicating a paper from the Jakuski lab from the University of Chicago. And what we found was that at this suboptimal dose, while animals from one vendor were able to respond to the checkpoint inhibitor therapy to have inhibition of tumor growth, the animals from the other vendor we're already sort of at the low level of that the taconic animals ended up as, so that they couldn't respond anymore to the checkpoint inhibitor therapy. And this just shows that the microbiome allowed us to start in a different place. So the microbiome of the taconic animal was able to be was educating the immune system such that we were able to inhibit tumor growth with the checkpoint inhibitor therapy, while the JAK animals were already had that extra anti-tumor activation of the immune system, and so the checkpoint inhibitor therapy wasn't able to do much there. And this really provides a nice framework for a model where we can, using this same paradigm, look at differential contributions to therapies that might have a microbiome modulating piece to it, and allow us to either see a response or not, and really look at the microbiome contribution to that. We also ran this in a germ-free setting, and just to show you that data really quickly, the germ-free animals, the tumors in the germ-free animals grow even less. And this is more of an academic question, and looking at how the germ-free respond. Their immune system has not been educated by exposure to any previous microbes, and for reasons maybe not totally understood at this point, their tumor growth is less, and obviously they don't respond to the PD-L1 therapy either. All of our studies have daily weights as a part of the therapy every day, so I'm just going to show you this one graph of the daily weights from that study. This is a bit of ex vivo data showing some flow panels from the tumors, looking at tumor infiltrating lymphocytes. We have the ability to look at a wide variety of cell types and distinct populations, also to sort or deplete specific populations, and run a wide range of immunoassays to look at various endpoints. These include early spot assays, MLR, reactions. We're able to do multiplex cytokine analysis with our Luminex instrument. We're able to do qPCR looking at gene expression, et cetera. So not to get too much into the data here, it's more just to show you a bit about the potential options for ex vivo assays there. Additionally, we really think, and we've really found that just looking at tumor lymphocytes is only going to tell you one part of a really systemic system that we're looking at. So when we want to look at the relationship between health and disease, we want to probe from multiple sites to really get an idea of what's going on locally. So we might do ex vivo assays looking at lamina propria or other sites. We've also done studies looking at oral microbiome, but looking at the local interface between the microbiome and the immune system, also at more systemic places of interest like the spleen or lymph nodes, and then at the effector sites where the actual tumor killing or immunosuppression is going to be happening at the site of the tumor. I want to introduce really quick the concept of the Oncobiome. So in oncology, whether through disease or through treatment, the microbiome may be altered into what's been termed the Oncobiome. This is basically the patient's altered microbiome in the context of oncology. And we can look at customization of models to really look at the oncobiome. So we can induce an oncobiome also in a tumor context via chemo, radiotherapy, or immunotherapy, or antibiotics, or introduction to specific pathogens, and then run our tumor models and or microbiome readouts to assess these important clinical endpoints. I also want to mention that we can, I mentioned it really quickly before, but we're developing transplantation of clinical microbiome, whereby we use the germ-free mice. We would introduce a clinical either responder or non-responder. You can have disease or healthy, different microbiomes into the mouse, and then breed the mice, allow the F1s to grow up so that their immune system has been completely educated by this specific human microbiome that's going to be stable, and then assess readouts using the F1 mice and perform the models with various overlays. And of course, a number of in vitro assays as well to look at a whole host of different microbiome and immune-related endpoints. So to summarize this first model paradigm for customization, this is the microbiome in immuno-oncology. Microbiome is a key mediator of anti-tumor immune response, should be acknowledged and controlled for in assessing efficacy of all IO therapies. Biomodels expertise in custom and rationally designed preclinical models can be applied to assess specific microbiome-Oncobiome-associated therapeutic targets in clinical context. Biomodels' Gnotobiotic germ-free facility offers the opportunity for IO studies in the context of clinically humanized microbiome, as well as many other applications. I didn't mention this specifically, but we've benchmarked a wide range of murine cell lines and established minimally effective suboptimal doses for a number of the checkpoint inhibitor antibodies to enable combination studies with those antibodies. And we offer a wide range of ex vivo assays to enable biomarker identification, confidence, and mechanisms of action. These include multiplex with Luminex, FACS, including sorting, microplate assays, LA spots, et cetera. And we have the experience in working with various adjuvants, sites of administration, local systemic, and performing isolations of various organs of interest, looking at antigen profiling of tumor lines, et cetera. A lot there. And this is just a brief and probably incomplete list of some of our murine cell lines that can be utilized in these kinds of models. These can also be overlaid with genetically modified mouse models to look at very specific impact potential targets in a number of different ways, or potential reporters, combine a number of different schedules, or overlaid with related indications. So moving to the next kind of case study, this is going to show a bit about modeling standard of care chemoradiation regimens, relapse orthotopic sites, bioluminescent reporters. So this, we're going to talk about a model of glioblastoma. Obviously, it's a pressing unmet clinical need for this devastating indication. Normally, we would expect to see disease recurrence following initial response to a standard of care treatment. And so the goals for preclinical modeling in this case is to have the accuracy of context and an intact immune system we're going to try to model IO therapies and to model the relapse recurrence. So I'm going to talk now quickly about an orthotopic bioluminescent model of Murine glioblastoma. This uses GL261 cells that express a luciferase reporter gene, and we apply these cells intracranially into the brain of the mouse, and we can follow over time with in-life imaging using our IBIS instrument, and overlay it with standard of care temozolomide or temozolomide plus fractionated radiation, which is probably what a patient might receive. Obviously, a very strong readout would be survival. Often studies are organized such that we can capture either survival or a predefined endpoint to look at more ex vivo readouts. But if you want to do survival or both at the same time, but if you want to do survival, survival is obviously a very compelling endpoint. We can see here that the vehicle mice have much diminished survival compared to the temozolomide, mice receiving temozolomide, or those receiving temozolomide and radiation persist even longer. And this was even cut off. Here we can see some tumor growth kinetics in vivo using IVIS. So we can see here the actual signal from the brains of the mice. And importantly here, we can see relapse occurring. So we can look at the kinetics of the response and the relapse, and we can apply your novel therapeutic or a combination and really assess pretty accurately in real time. And here's another look at weight change from the study. So this is a quick slide just to show you a few of the places we have expertise in seeding tumor xenografts or allograft cells. So we can seed in a number of spots, and some of these are more tricky than others. Surgical implantation of tumor cells to various sites does require some expertise, and we do know how to do it for a variety of sites here at Biomodels. So to summarize this portion, modeling standard of care chemotherapy regimens, recapitulation of clinical standard of care treatment, relapse is going to be key for translational success. Recapitulation of tissue context is also going to be key. We have expertise in administration of tumor cells to a wide range of tumor sites. We have expertise with a large number of bioluminescent or fluorescent reporters. Using in-life imaging enables accurate assessment of in vivo tumor kinetics and allows for precise endpoint ex vivo assays, and also for metastasis studies, which I'll touch on in the next section for a minute. Biomodels has expertise in application of targeted radiation, chemoradiation schedules, et cetera, to enable clinical recapitulation. And I talked a bit about survival studies versus scheduled endpoint studies. So next, we're going to quickly move to our bioluminescent PDX, focus on metastatic models and more clinical recapitulation. Through a partnership with Zentek, we're pleased to be able to offer well-characterized bioluminescent tagged PDX lines. I'm going to talk to you today about, I'm happy to talk more offline about how these are generated and a little bit more about them. But I'm going to talk today about a triple-negative breast cancer and a pancreatic cancer bioluminescent PDX line. We've characterized these with principal component analysis, and we know what the difference is in terms of putting the luciferase cassette in, and the differences are not that great. It's similar to the differences between orthotopic and ectopic sites in terms of gene expression, and we have the data showing which genes are affected. And it seems like these are very good, true models, even with the luciferase reporter placed in there. And the luciferase reporter, of course, gives us a whole bunch of extra power in terms of looking at metastasis and following in vivo, but also does come with some considerations in terms of immunogenicity and those gene expression considerations that I was just talking about. So I'm going to talk quickly about, this is an orthotopic bioluminescent PDX model for pancreatic cancer. Again, a very pressing unmet clinical need. Accuracy of context is going to be critical. We're going to need an intact immune system for IO therapies. We want to model disease recurrence. In this model, we use NSG mice, so no immune system here, but we're using the human PDX tumors. We're using targeted radiation or standard of care gemcitabine or an experimental, more experimental JQ1 treatment we're able to follow over time, and we're able to assess local metastasis and look at survival of those readouts. In the triple-negative breast cancer line, this is more of a metastatic model where we're looking at treatment immediately following surgical resection, which would be the most common treatment paradigm for a patient. So the tumors are allowed to grow up, you can see on the top right, and then the tumors are resected, and the mice are enrolled one by one at a specific tumor radius. And as they go on therapy, as they're enrolled, they go on to therapy, and they're administered either radiation therapy or various standard of care or experimental therapies. So you can see in these in the mouse images here that we've resected the primary tumor, and we're assessing here in the graph at the lower left just the metastatic progression, so just the lung signal at this point. So to sum up this part really quickly, so I told you about our panel of bioluminescent PDX and how these can be used in metastatic models that are clinically recapitulative. Again, recapitulation of clinical standard of care treatment and relapse is going to be key for translational success. Tissue context is again key. We have expertise in administration of tumor cells to a wide range of tissues again, and our expertise with reporters, imaging, and I already said that, in vivo tumor kinetics allows precise endpoint ex vivo assays and test studies. We can apply targeted radiation on specific chemoradiation schedules to model the clinical situation, and also test in surgical or neoadjuvant settings. So next I'm going to talk about cancer treatment toxicities in tumor context, simultaneous modeling of related indications. Sorry about the picture, but we're going to talk quickly about mucositis. This is a prototypical cancer treatment toxicity. It's seen in nearly all patients receiving therapy for head and neck cancer. It's impossible to segregate the supportive care from the tumor therapy. So if you have a treatment not only against an anti-tumor treatment itself, but against a cancer treatment related toxicity, it really needs to be assessed in a tumor model. So we've developed preclinical orthotopic head and neck cancer models designed to co-assay therapeutic toxicities in the context of tumor growth and treatment. So we have these for oral mucositis, and we can also do this in xerostomia. So this is just the model setup again. So we're using nude mice in this case with a bioluminescent tagged head and neck squamous cell carcinoma cell line, SCC25. And we're going to be able to perform mucositis evaluations and also tumor growth evaluations using the IVIS. So here's just a quick summary of the data, and I'm going to kind of just push through this and let you read this later. But basically on day 18, we're seeing tumor growth and decreased tumor growth in the targeted radiation group, whereby in the targeted radiation group, we get increased mucositis score versus the untreated group has no mucositis. So to summarize this section, cancer treatment toxicities that are intrinsically related for comorbidities that are intrinsically related, preclinical testing must be performed in the context of both indications. Biomodels offers overlapping models to enable evaluation of cancer treatment toxicities, mucositis, xerostomia, chemotherapy-induced diarrhea, for example, in the tumor context. And additional models are also under development, such as GVHD and GVL, graft versus leukemia situation, antibiotic immunosuppression, et cetera. So last, I'm going to talk about sort of next generation models, referring to as triple-humanized models. Also talk a little bit about scheduling and tumor microenvironment in vivo. So to go back to pancreatic cancer, the tumor microenvironment is known to be of extreme importance in pancreatic cancer. Not only do we have a lot of immunosuppression happening as a result of cells that are really bone marrow derived or elsewhere forming immunosuppressive or actually physical barriers to drug delivery. And one of the key cells that has been recently sort of identified as playing a role in both of these, as well as in many other cancer contexts, are the mesenchymal stem cells, or the MSCs. So what we wanted to do here was to triple-humanize a mouse, whereby we have a mouse with a human tumor, in this case a BXPC3 pancreatic cancer line expressing a bioluminescent luciferase reporter. We also we're putting these in with into the pancreas, where we're following with IVUS. We're also humanizing the immune system of the mouse with peripheral blood mononuclear cells from a previously characterized donor, and we've worked out this donor for response to checkpoint inhibitor PD-1, and also we've worked out the numbers so that we're not seeing much or any GVHD as a result of putting in these cells, which is definitely one of the limitations of some of these humanized models. And finally, as a third piece, we've added co-implanted human bone marrow-derived MSCs, and these are actually given as bolus doses once a week to simulate translocation of these cells from the bone marrow out to the local tissue sites where they have been shown to home to tumor sites. And the idea was to assess the effect of the MSCs on the efficacy of the checkpoint inhibitor therapy. The first thing we wanted to do was to measure engraftment, and we did obviously a lot more than I'm showing you in any of these case studies, but this is just looking at peripheral blood CD3 positive cells. And this is where we found a very surprising result. We found that we had engraftment regardless of MSCs when we put in the PBMCs. However, when we treated early with the anti-PD-1 antibody, the antibody treatment completely, almost completely prevented engraftment. So this was an interesting result, and it agrees with some of the other literature coming out at the moment. We're going to be presenting this at ACR this year. If you're going to be there, please stop by and say hi. But it really, we think, has some clinical implications too in terms of scheduling of immune checkpoint inhibitor treatments with potential various effects that might not be intended. So important for scheduling there. This is our total tumor radiance from the animals in vivo. You can see that administration of the PBMCs really suppresses the growth of the tumor cells as would be expected. And we really didn't see that much of a contribution from the MSCs, maybe a little bit. You can see here in the ex vivo pancreases, maybe we have a little bit of an increase, but really the MSCs we would expect more of an increase on metastasis and not primary tumor, and we did not have the opportunity here to assess metastasis. We perform much of our histology work with our partners, Histotox Labs. They do great work in terms of a lot of IHC, and also are able to quantify. So this is some quantitative IHC from the tumors, just looking at CD45 positive cells. And again, you can see for the PD-1 treated animals, we completely prevented engraftment altogether, whereby in the MSCs, we did get a somewhat of a decrease in the mean tumor infiltrate there. And then just a few more flow readouts here. So just to summarize this part, we're constantly innovating, trying to innovate and develop new models. Triple-Humanized models in this fashion, and also in the clinical FMT that I mentioned earlier are poised to add new levels of accuracy to preclinical modeling. They're opening up, hope for them to open up efficacy testing of new tumor microenvironment components, especially MSC's contributions, myeloid cell contributions, et cetera, in humanized contexts. And we're developing more of these, as I just mentioned. And I'm just going to stop and summarize altogether by saying that development of novel anti-cancer therapeutics remains an urgent clinical need. Development of novel anti-cancer therapeutics is more difficult than other indications, confounded by the individuality of the disease and treatment, and a continued dearth of clinically actionable biomarkers. Preclinical models can be rationally designed and customized to assess key translational properties in models with increasing biological accuracy, looking at mechanism of action, biomarker schedules, combinations, et cetera. And looking at all these things and our expertise here at Biomodels in the design and execution of these really highly custom preclinical models in oncology can help enhance translational success. So I'd like to thank everybody for listening today very much. I'll leave you with probably the most popular quote from JFK, but it's a little bit of a mantra for us. And we don't do things because they're easy, we do them because they're hard. And we want to do the things that are hard here at Biomodels well. We're happy to do your more straightforward models and studies as well, but we really want to focus on the custom and the hard preclinical models that may be ultimately providing more translatable and translational success. So thank you very much again from all of us here at Biomodels, and thanks to Cambridge Health Institute and Elizabeth as well. Please do contact me if you have any questions. I'd be happy to talk over your project and to talk more in depth with you about any of the things I glossed over in the talk today. Thanks a lot.
Thank you very much, Dr. Cuffo. I will plan on forwarding the questions that we've received to you via email as we're coming up on the hour. So I'd like to very much thank you for your presentation. I'd like to thank the folks at Biomodels LLC for sponsoring today. And I'd also very much like to thank those of you who came and attended today's webinar. We hope you gained some information that will make your research life a bit easier. So with that, thank you again to all of you, and have a great day.